Hello everyone. I'm JJ. I hope you're all doing really well. Today I made few changes. I hope you liked it. And today we'll start a new thing which is so big and so important. So without further ado, I'm welcoming my friend Ta-da! DNA. So years before, when DNA was literally playing hide and seek with us, and no one really knew that there was something inside our body, inside the cell, which could be actually transferred from our parents to us. At that point of time, so many scientists worked on this particular thing and finally got him. And that was none other than DNA. So let's meet those superheroes first. So the first person who actually found our DNA was Frederick Mischer, but accidentally, which means all accidents are not that bad. He was actually searching for some proteins in white blood cells and accidentally found DNA. Yeah, which he then named nuclein, which was later changed to DNA by some other scientist. So after that, so many scientists came in and tried to find what it actually was. And then came a bunch of superheroes, which included Morris Wilkins, Rosalind Franklin, James Watson, and Francis Crick. All of them were trying to find out how DNA looked. But it is said that Rosalind Franklin, that is this particular lady in the middle, was the first one to deduce the double helix model of DNA using a technique known as X-ray crystallography. On the other hand, Morris Wilkins was also using the same technique, that is X-ray crystallography, for the deduction of DNA. It is said that they were not in good terms, but we will not dig deep into that. So, at the same time period, James Watson and Francis Crick, who were also working on DNA, got this information, that is, that Rosalind Franklin got a result from her experiments. So, they took the reference of that information and published their first article in a magazine known as Nature, which got really great recognition. And further, Morris Wilkins, James Watson, and Francis Crick got a Nobel Prize for the same. But sadly, Rosalind Franklin was not included because it said that she passed away due to ovarian cancer. So that's a sad thing, I know. But yeah, she is still acknowledged for her great achievement. So that's a whole of a history, and now get into the structure of DNA. So these two things that you see, they are the backbone of DNA, and it is made up of phosphate and sugar. So this particular thing is made up of phosphate and sugar, and this latter sort of thing is made up of nitrogenous bases. So what happens is that phosphate and sugar is present here, and the nitrogenous basis which is attached to this just bulges out. Okay, so when they come together, they almost looks like this is the family picture. Just not, they are not smiling, I know that. I will give you a clear picture of how they really look. So this is the phosphate I was talking about, and then there's ribose sugar, and then there is our nitrogenous bases because there are nitrogen in this particular structure, right? And then this is the sugar, and this is the phosphate, which is triphosphate. There are one, two, and three phosphates, right? So we will first meet our busy guy, that is ribose sugar, which is busy making DNA. So this is how the linear structure of ribose looks like. So ribose is a pentose sugar. Pentose means it has 5 carbon. Okay, so we'll count it. 1, 2, 3, 4, and 5. Ta-da! So this is a pentose sugar with 5 carbon, and it is known as ribose. So can you give an example of a hexose sugar? What do you mean by hexose? Hexose means it includes 6 carbon, right? So can you give me an example of that? You can think that. So this is a pentose sugar, and we don't want a linear ribose in the case of DNA. We want a cyclic ribose. You have seen that here that it is a cyclic form of ribose, not a linear form. So how do we make it? For making a cyclic form, what we do is that the fourth carbon's OH group, from the fourth carbon's OH group, the O reacts with the first carbon and also transfer the H group to the this O. Okay, so here now it will become OH, and here it will be just left with a single O, right? So that's how it forms a cyclic structure. We'll just check in both of the structure. So this is the first carbon here, which has a H group and a OH group. Is it here the same? It is attached to a H group and of course a OH. The H here we got it from the O of the fourth carbon, right? So it is a OH and H, so we got that here. Okay, it's correct. And the first carbon is attached to the fourth carbon's O group. Okay, yes, it attached to the fourth carbon's O group, and the fourth carbon is attached to a H group and CH2OH, is it? So yes, the fourth carbon is attached to the H group, and of course the fifth carbon CH2OH. So it's correct till now, and then we are left with just 2 and 3. They both have a OH group and a H group. Okay, it's also correct. So at last we got a cyclic structure of ribose. So can we use this structure to form a DNA? Do you think it is possible? Just think. Okay, I'll tell you. No, we cannot use that because in DNA we need deoxyribose. As their name suggests, it is deoxyribonucleic acid. So it is a deoxyribose that we need here. So what we do is that we deoxygenate our second carbon's OH group. So that's how we get just H. We are left with just H. Here, in the case of ribose, there was OH, and when we deoxygenate it, we get deoxyribose with just a H group. So you have to remember that we are deoxygenating the second carbon's OH group, and that's how we get deoxyribose. So what do we do now? Can we throw the ribose sugar because it's not of use? No, we'll not throw that. Ribose is the main ingredient in the formation of RNA. So, as in the case of deoxyribose, that is, when DNA is made, this particular sugar is used, right? Here, deoxyribose is used. In the case of RNA, here O is present, that is, ribose, simple ribose sugar is used. So, here there is no waste in this particular slide, so we would use both of the sugar. Yeah. And now we will learn about the second ingredient, that is what? Yes, phosphate. So here I said that is there are triphosphate, that which means three phosphate group is present. But how? How that does that happen? So what happens is phosphoric acid is the one which is present over here. So three phosphoric acid comes here and forms a triphosphate. So this is how phosphoric acid looks like, okay? And when they come and attach, it forms a bond with sugar. We'll see how. So this is the fifth carbon of what? Deoxyribose sugar. Okay, here it was CH2OH, the previous slide. I just shifted the OH group from this side to this side so that it could be more clear for you. Okay, and the phosphoric acid already has a OH group, right? So it has a OH group over here also. So the OH group and the CH2OH group of the sugar comes together, and that's how H2O are released. Okay, so let's see how. This is one H, this is second H, and there's one O. So H2O. So H2O is released and a bond is formed. The bond's name is Ester bond. Okay, you have to remember that. And ester bond is a bond which is found between acid and alcohol. So here phosphoric acid is used, right? And then alcohol because a OH group is present. So here we got an ester bond and H2O is released. So the first phosphate which is close to the fifth carbon is known as alpha carbon, okay, as it is close to the fifth carbon. And then comes beta because it's little bit far, and then comes gamma which is so far. So that's all about the phosphate group. You just have to remember about the ester bond and also the release of a H2O. Okay. Now let's discuss about a third ingredient, and that is none other than nitrogenous bases. So they are divided into two teams, that is team purine and team pyrimidine. So the thing is, team purine has two big members, that is adenine and guanine, and the most surprising thing is that they are friends with their opponents, that is team pyrimidines. So what happens, or what is the term when one base pairs with another base? So these things are bases, right? They are nitrogenous bases. So what is the term we use when a base is paired with another base? It is known as base pairing. So adenine always base pairs with thymine, whereas guanine always base pairs with cytosine. You can see that here. If this is our DNA, this is our DNA, okay? And these strands is occupied by sugar and phosphate, which are bound to each other, right? So the bases bulges out, and that is here we can see that A stands for adenine and T stands for thymine. So A and T, they are base pair together, and G and C, that is G and C, are together. The same happens, it can also happen in opposite manner. A is here and T is here. So, but the cytosine cannot make a base pair with adenine, nor can guanine base pair with thymine. If that happens, then there is a problem. Okay. So there is another base pair, that is uracil. So why is it here? The thing is, uracil can also base pair with adenine, but dear thymine, you don't have to worry because adenine is all yours, because uracil is not present in DNA but just in RNA. So we'll see how adenine and thymine makes bond. Okay, so they have two H bonds which keep both of them together, and then we have guanine and cytosine with three H bonds. So whom do you think is stronger? Adenine or thymine with two H bonds, or guanine and cytosine with three H bonds? Of course, guanine and cytosine, right? So that's why they got the best pair award. So the thing you have to remember is that purines always have a basic structure. That is, they have a six-membered ring and also a five-membered ring. Okay, you can see that here in guanine. Okay, I'll show adenine also. Adenine also has a six-membered ring and a five-membered ring, right? So that's the basic feature of purines, and that's why purines are huge in comparison to pyrimidines, which just have a six-membered ring. So this is the ring of cytosine, and then there is thymine with six-membered ring. So that's how it works. Only difference is about the functional group which is attached on these rings. So in the case of adenine, if the numbering of the adenine starts from here, okay, so it is 1 2 3 4 5 6, we have amino group on the sixth position, right? But in the case of guanine, what happens is that there is oxygen on the sixth position, right? So there are a lot of differences in between them on the basis of the functional group. So in general, the purines numbering start from this nitrogen. So it is 1 2 3 4 5 6 7 8 9. Okay. And then in the case of pyrimidine, the numbering starts from here. Remember, in the case of purine, numbering starts from here. In the case of pyrimidine, numbering starts from here. So it is 1 2 3 4 5 6. So here we can see that there is a amino group where on the fourth position, right? 1 2 3 4. But in the case of thymine, we have oxygen on the fourth position. So that's the difference. Okay, so you have to learn that, you have to see the functional groups because it is really important. So now we'll jump onto sugar bonds with a base. So this is a base and this is a deoxyribose sugar. So I have taken example of a purine, right? This is a G, that is guanine, and guanine is purine. So guanine's ninth nitrogen has a H group. Of course, every nitrogen has a H group over here. I've not mentioned it because it is not necessary, but you have to assume it that there is a H group. Okay, so here I have mentioned H because it is of use. So we will see how. So this is the ribose sugar, deoxyribose sugar. So on the first carbon, OH group is present, right? So the OH group interacts with the H group, and that's how H2O is released. And this particular bond is known as glycosidic bond, not James Bond. I know it's a bad joke. I have written that also. So what happens? There a bond formation occurs, and this bond is known as glycosidic bond. So there's an important thing that you should know, and that is when a sugar and a base come together, it is known as nucleoside. This whole structure is known as nucleoside. Whereas when a phosphate is also included, that is a phosphate, a sugar, and a base, when that is included, it is known as nucleotide. So DNA is formed of many nucleotides like this. Okay, so DNA is formed of one nucleotide, second nucleotide, third nucleotide, and that's how DNA is formed. We will see that in the next class. I'll just finish about the basic things over in this class, and then we'll see how this particular structure is formed. Okay. So we have seen that a glycosidic bond is formed in this case, that is in between a sugar and a base. Okay, you have to remember that, and an issue is released, and this particular structure is known as nucleoside, not nucleotide. Okay, nucleotide is only when a phosphate is also included. So, in the case of pyrimidines, the N1 nitrogen is attached to the first carbon's OH group, whereas we have studied, right? We have studied in the case of purines, the N9th was attached to the first carbon's OH group, and hence H2O was released. But in the case of pyrimidines, N1 is attached, and then of course H2O is released. I have not shown here, but yes, H2O is released. So I have put a note over here that is N9 for purines and N1 for pyrimidines. So that's how a glycosidic bond is formed, and then of course this is known as a nucleoside, but when a phosphate comes it is known as what? Yes, nucleotide. So many such nucleotides come together and form a structure that is known as DNA. So if this is one nucleotide, the second nucleotide comes, and that's how a DNA is formed. So I will teach that in the next lecture. Until then, I thank you for bearing with me, for cooperating, for watching my video. So if you like my video, please give us a thumbs up and share and subscribe. Thank you. Much love. Bye from JJ.